Welcome to Oncology Data Advisor. Today I have the pleasure of being joined by Dr. Jesus Garcia. Thanks so much for coming on today.
Thank you.
Would you like to introduce yourself and share a little bit about what your work and your research focus on?
Sure. So I'm originally from Mexico. That's where I went to college. I originally came to the United States for grad school. So I did all my research in collaboration with the Houston Methodist Research Institute. I rotated through different labs, but I ended up in the pathology lab there. So after graduating my PhD, I got the opportunity to work at MD Anderson Cancer Center as a scientist there in one of the laboratories under the umbrella of Dr. James Allison, which was a privilege there. It was the immuno-oncology platform over there. That's where I started learning and developing skills as a scientist, specifically in the biomarker space, working in already clinical trials or assays for clinical trials. I dove into new technologies, not only like immunohistochemistry and multiplex immunofluorescence, but I got in to learn about image analysis, digital pathology, and more. After a few years there, I switched into industry, into a small laboratory called Apocell, which pretty much provided lab services for clinical trials, mainly for biotech or biopharma. And it was mainly specialty lab services around tissue and liquid biopsy assays. So I was a scientist there for about a year or two when I transitioned into a more of a subject matter expert role. And that's what I've been doing for the last 4 or 5 years. So pretty much my role in my research, or the part that I play in the research we do at Precision for Medicine, is to really dive into the sponsors' clinical protocols and see how we can help them develop an appropriate biomarker strategy. Now, even though it's not exclusive, the majority of our projects are oncology, and a big part of that is immune oncology. So that's why a lot of my work has been in the tumor microenvironment, for example. So that's a short introduction, I guess.
Awesome, thanks. I'm excited to hear more about this research too.
Yeah.
So for background, what is the tumor microenvironment and what is the role that it plays in tumor growth?
Sure. So historically, we have been very simplistic when approaching cancer, right? We have been developing therapies, and I'm talking some time ago. It has been a very simplistic approach, taking into account just understanding a tumor cell and how it responds to X or Y therapy. That's why we came up with different, more targeted, like not necessarily targeted therapies, but therapies that were not taking into account cancer or tumor as a whole, as a complex system. Okay, so that's when research starting gearing towards, okay, why are a lot of these therapies failing? And then we started trying to understand that the tumor is not an isolated system. It exists in a more complex environment, the tumor microenvironment, where there's a lot of things that play a role. Some of those things can be different proteins like cytokines, or things like how much oxygen is present, the pH or acid involved in the tumor microenvironment, and obviously a lot of different potentially impactful cell types. Not only tumor cells, since that's what we've been focusing more for the last couple decades, but also other cells like cytotoxic immune cells, suppressive immune cells, and different subtypes of these cells. So we've learned to look at the tumor and the tumor microenvironment, some sort of super Darwinian ecosystem where survival of different clones of these tumor cells matters, and how they survive to their environment, to maybe even therapies or the immune system. So that's, a little bit of what the tumor microenvironment is everything around the tumor involving other cells, necrosis around the tumor, blood vessels around the tumor. And all of that is going to have an impact on whether, not only whether the therapy is effective, but whether it's delivered, whether it's going to have unintended consequences, etc.
Great, thank you. That was a really helpful overview in understanding it.
Thank you.
So what are some of the current unmet needs regarding understanding the tumor microenvironment?
Well, one of the main things is that the tissue availability is very, it's always an issue, right? That's because obviously, in order to obtain tissue to analyze it, a patient has to undergo an invasive procedure, right? And some of those can present themselves potentially a significant risk depending on where the tumor is, and the adjacent structures. Lung biopsy can be very, very dangerous just by itself. So it's always a challenge to obtain enough tissue to explore the tumor microenvironment. And then there's limitations of technology. Immunohistochemistry is one of the gold standards that have been used by pathologists and clinicians. But that, even though that's still the gold standard for many purposes, it's very limited, and it's potentially wasteful, because you're analyzing, you have one analyte, and so just you're looking at one protein and you're using one whole slide. And depending on the size of the tissue, you might have just a handful of slides available for you to get a better understanding of the tumor. Now, even though it's still very useful, a limitation has been on, okay, how can we make better use of the limited amount of tissue that we sometimes have? So even though we're making great progress, now we have, speaking again about protein, for example, we have multiplex immunofluorescence approaches where now we're able to look at many more proteins at a time. Now, this is not, immunofluorescence is not a new technology, but the quantity, the number of proteins that we can look at a time, the sensitivity of the assays, the throughput, just the quality in general of this technologies, it keeps increasing. Now we have multiple technologies where you can, in theory, measure hundreds of proteins, right? It's like doing 100 IHC assays in one slide. Now, the challenge that presents then is data analysis. Before, scoring an IHC slide was relatively simple. Even for a pathologist, of course, they have to look at a handful of areas, quantify the expression of these proteins, see how many cells, assess the intensity of the expression of this protein, and then come up with a scoring, whether it's a H-score or a custom scoring system. But it's relatively simple. Now, once you turn that into, okay, now we have, not only one, but 2 or 10 or dozens of proteins in the same slide. We want to do the same thing, but on a different scale. And it's not only like doing the same thing, times 20, times 30, but now we also want to assess, okay, now that we have those 30 markers or whatever, how do they relate to each other. Now we can define potentially more complex phenotypes of cells. Okay, and before we could say, okay, we have, this score for CD8 cells. Now we can see, okay, we have a CD8 cell, but that is expressing this marker X, Y, and Z. And now we can do the same as assess those cell types and in context of other complex phenotypes. So it really becomes not only the complexity of the data and the analysis that needs to be done, it's exponential. It's not like analyzing 30 IHC, scoring 30 IHC slides. The complexity of the relationship among all those different proteins or cell types becomes much more complex than the sum of 30 proteins. So that's one of the main limitations right now. We're starting to come up with, by way of the field is starting to come up with solutions on how to even ask the right questions, because when you have just a handful of proteins to analyze, it can be not as difficult to ask a question. Okay, how many CD8 cells have, how much intensity I have. But once you have all those 30 markers or more, then even phrasing a meaningful question, and that is going to be useful for your patient ultimately, it becomes a real challenge. So I think that's the main, that's one of the main limitations right now. And same goes for genomics-based assays. Genomics produces a lot of data, but now that we're also starting to use the NGS or other molecular tools to analyze the tumor microenvironment, in the context of in the spatial context, because we have always done bulk sequencing or whatever, but now we're starting to add the spatial variable to it. Same thing happens. Like now that we're sequencing all these different cell types and we're trying to take the variable of spatial context into account, then it just becomes exponentially more complex. So those are probably the main limitations right now.
Great, thank you. So in light of all these complexities and the solutions that are being made to address them, how can an improved understanding of the tumor microenvironment impact oncology drug development and ultimately patient outcomes?
Where we can see that understanding the tumor microenvironment can be more impactful is in immunotherapy right now. That's the textbook example right now, because of course immunotherapy needs it uses the immune system, right, to treat the cancer. So in order to understand how the immune system is going to respond once the therapy is given to the patient, we need to understand the context, the spatial environment, the spatial context of this tumor, right? We typically, the main three categories, for example, that we see or that we use to classify the immune infiltration in the tumor is like either hot or cold, or but it's really a continuum, right? It's not like it's hot or cold. Is the degree of immune cells involved in this region in the tumor microenvironment, or the lack of these immune cells. So obviously, if we see that there are no immune cells in our therapy needs immune cells, we either need to recruit immune cells to the tumor, or decide that this therapy might not work for this patient. Sometimes it's even helpful to, we're not even going to select this patient for this therapy because it's very unlikely that it will work for them. So there's not only, of course, we can save costs, but also like potential secondary effects that might impact the patient without actually giving them any benefit hopefully, so they can probably move on to the next possible alternative. So understanding mainly the immune cells, that's the easiest example, where they are, if we have the target phenotypes that we are interested in, because immune therapies are given for can target different proteins or different cell types. They might want to involve more regulatory cells or more cytotoxic cells or other cell types that might be involved, like macrophages. So we have to understand, okay, how many of my cells of interest are there? Where are they? Because if they are outside of the tumor, if there's no infiltration, we might need to say, okay, maybe it's not going to work because something is preventing the immune cells from actually working where they should be working. Okay, but if we see the right conditions, if we see that either before or after therapy, we can see that, okay, we have the correct cell types, we see the protein and the targets of interest in the appropriate regions, then we have a better chance of well, the patient might respond better to these therapies. So in our case, what at Precision, what we're hoping to help sponsors with is first developing assays that they're going to help in their exploratory endpoints at the beginning. So they're going to mainly provide this type of information or data so the sponsors can learn or it can help the sponsors explain the results of what they are seeing after therapy, after the whether it's correlating with survival, with other measures. So we're trying to provide them with that information, and hopefully even if things go well in the trials, hopefully we can help them develop assays that are going to help them predict, right, to really help them achieve this precision medicine, to help them come up with the appropriate subjects or patients that are more likely to respond to their therapies. So this is one way of how assessing the tumor microenvironment can really help with precision medicine.
Awesome. So speaking about these trials that are underway or that are planned, what is some of the progress that you hope to see in this field within the next few years?
Right now, of course, there's a lot of traction with the typical checkpoint inhibitors, the PD-L1, the CTLA-4, etc. But for the typical therapies, that the first couple of checkpoint inhibitor therapies, the tumor microenvironment was not assessed as with the technologies that we have right now. So right now we see a lot of the sponsors trying to use these technologies to improve therapies that have already been doing very good, but have somehow failed to provide the response that we were expecting at the beginning, at least with some indication of cancers, right? Right now, your typical PD-L1 therapy, we still use a companion diagnostic that is just a single chromogenic IHC, so one target, and the way we score that is relatively, still old-fashioned, which, don't get me wrong, is still gold standard, but it still lacks the tools or the technologies that we now have available. So I see a lot of clients or sponsors trying to just use that technology, maybe multiplex immunofluorescence. Maybe they want to understand not only PD-L1 expression in tumor cells, but how does that relate to also regulatory cells around the tumor and cytotoxic cells around the tumor? So trying to use more complex ways of selecting patients that are more likely to respond to the therapy, which it's already good just by doing an IHC, but if you use the current technologies to do a better selection of those patients, we're more likely to have better results. There's also HER2, from ASCO, I think it was last year, there was very good advancements with the low expression HER2s that previously they were classified as negative, but a group made some awesome advancement there where they saw that some low expression, some patients that had low expression of HER2 could potentially still get benefit from targeted HER2 therapy. And now also, again, using better technologies or more advanced technologies from digital pathology and everything, even in just IHC, another thing that has been implemented is helping the pathologist with the scoring system. So there's some companies that have used AI, artificial intelligence, to help the pathologist with better scoring. Okay, because for example, the pathologist might be focusing on just a few fields of view to score, while the AI algorithms can just do it the entire slide. Sometimes AI can distinguish smaller differences in expression or counts, or they can count the entire, like every single cell in the slide, versus the pathologist that relies on a smaller sample. So there have been some studies where retroactively they say, okay, if we had used the AI to help the pathologist to enroll patients, they would have enrolled more patients that were going to respond to the therapy versus what they originally did, which was enroll with the pathologist scoring method. So we're already seeing those improvements and how using more technology to better assess the tumor microenvironment is going to help with mainly immunotherapies, but not limited to.
Great, thank you. There's another really great overview of all the progress and the upcoming advances. So anything else you would like to share on this topic?
No, I mean, just thank you so much for having me. This is a great opportunity, and I always love talking about what I do, about not only cancer, but all the advancements in the tumor microenvironment, digital pathology, even AI, and how it's helping us come up with better treatment for patients. I think I always like the opportunity to discuss and talk about these things. So I just wanted to say thank you for having me.
Yes, absolutely. And we look forward to hearing more about the research in the future as well.
Awesome. Thank you very much.